In our next lesson on membrane transport from Chapter 9, we want to look at selective channels. Ion channels are more complex in structure than porins, and this allows them to be more selective about what they transport. Remember, porins are mostly beta baryls, but ion channels are multimers of alpha helical segments, and that's illustrated in our figure on the right here. The ion passageway, or pore, is in the central axis where the helices meet, highlighted by our blue arrow here. Let's look, for example, at the potassium channel from S. lividans. And that's illustrated in our ribbon diagram on the upper right here. It has 4 subunits highlighted here by the different colors, purple, blue, green, and red. As you can see, each monomer has 3 alpha helices, 2 large ones and 1 small one, and I've labeled them for you in the red monomer here. 1 helix from each subunit forms that central pore. One of the helices from each subunit faces the hydrophobic membrane interior, and we can think of that as helix number 2 here. And as you can see, that third helix is shorter and facing the extracellular side of the protein. We can see a little bit better how that pore forms by looking at the illustration on the lower right here. The potassium ions are purple. If we look on the right, we're looking down that central pore, the axis of that pore, and you can see our tetramer here. The monomers are highlighted in different colors, and I've labeled the 4 helices, one from each subunit, that contributes to the formation of that pore. Well, we say the ion channel is selective. How selective is it? The potassium channel is 10,000 times more likely to transport potassium than its cousin sodium. If we look at the periodic table, we find that potassium and sodium are in the same column. Sodium is just smaller. So the selective nature of this channel has to do with the geometry of what we call the selectivity filter. The pore narrows to about 3 angstroms, and that fits perfectly our potassium ion, highlighted here by the purple sphere. Each of the backbones for those 4 helices fold so that carbonyl groups project into the core, and that's highlighted in our figure on the lower right here. Those oxygen atoms in each case are highlighted by the red tips. It is those carbonyl groups, that carbonyl oxygen, that makes it selective for positive ions. Although there's no negative charge, we do have a very electronegative element in that oxygen atom, and that will make it more selective for positive ions. But what about the size? Potassium is a good fit, and sodium is too small. It can't coordinate with those carbonyl groups. You want to think of those carbonyl groups as so many hands surrounding that purple sphere to bring it inside the cell. The only way then that we could coordinate that smaller sodium atom is to move those carbonyl groups. We have to contract the pore. Here's where these tyrosine residues, and they've been labeled here, come into play. They're bulky, and they prevent the contraction of the pore to accommodate the smaller cation. It's as if I have my hands positioned perfectly, so if you throw me a nice big beach ball, I can catch it well. If, however, you throw me a baseball, I'm going to have to move my hands so that I can grab that smaller sphere. But in this case, I can't move my arms because of those tyrosine residues. And that's why it's so selective for potassium and 10,000 times more likely to transport that than sodium. Another example of a selective channel is an aquaporin. They were discovered by Dr. Agre in 1992. They allow for the rapid transport of water. Some water can move through the membrane by simple diffusion, but for larger volumes of water transport, we need these aquaporins, and so they're very common in nature. We're going to look in particular at the AQP1 family, and that's illustrated in the ribbon diagram at the top of the screen here. They are homotetramers, similar to the ion channel we just looked at. They have carbohydrate chains on the extracellular surface, but we won't be looking at that in any detail. In our ribbon diagram at the top of the screen here, we have illustrated the monomer. Each monomer is mostly 6 alpha helices that span the bilayer and 2 shorter helices that are within the bilayer. So you can see the colors here of those 6 alpha helices: yellow, red, orange, light blue, dark blue, and green. And here are the 2 shorter helices in red and green. Each subunit is a water pore, so we have a homotetramer. We have 4 water pores in each tetramer. The size is about 3 angstroms, and that is sufficient to accommodate a water molecule, which is about 2.8 angstroms. You can get a better idea of the size of that central pore by looking at the lower figure here, the ball and stick model, and there's our central pore within the monomer. Well, remember, we're talking about selective channels. So how could that pore be selective for water? They are lined with hydrophobic residues, except for 2 asparagine residues, which we'll look at in just a moment. Those hydrophobic residues are highlighted in yellow here, and I've labeled them for you. As you can see, there are 5 hydrophobic side chains that line that pore. At first glance, it might seem odd that we're lining a pore with hydrophobic residues to move through a very polar molecule like water. But remember, water readily ionizes. It readily picks up a proton to become H3O plus or hydronium ion. So if we're not careful, if we don't distinguish between the two, then when we transport a large volume of water, we'll transport a large volume of hydronium ion, and that will rapidly lower the pH and perhaps damage or interfere with some of our cellular processes. And so those hydrophobic residues prevent the transport of H3O plus, perfectly coordinating the structure with its function. The size of the pore is sufficient to accommodate water, so those nonpolar molecules don't prevent the polar water from getting in, but it does prevent the movement of anything charged. What about those asparagine residues? They're highlighted in red here. Remember, it has that amide group, and so it can readily hydrogen bond with water, and that allows us to transport a single water molecule. Remember, water is highly networked, and we want to, although we might want to move through a large volume of water, we want control. And those asparagine residues allow us to hydrogen bond and move through one water molecule at a time. Again, an excellent example of a selective channel. In both the ion channels we looked at and the aquaporins, remember, they're always open. They are just more selective than the porins we saw in the previous lesson. So in our next lesson, we want to look at gated channels and see how they differ. They have an open and closed conformation, and so we want to see what types of signals control these gates.